Welcome to geneXplain's webinar, COVID-19, Online Discovery of Prospective Drug Targets and Personalized Treatments with Genome Enhancer.
In the first paper, there were two patients which were analyzed. There was one patient which was analyzed, and the second paper, there were two patients. So in total, the 3 patient data. So for each patient, we will get different sets of drugs, which are shown here in this picture, the top 3. And of course, we have to tell, disclaimer here that these obtained results are fully computational modeling, and they need further validation before basically using this in clinical practice for treating patients. So it has to be taken with care, of course. So again, so what the algorithm of this tool is the following. So basically, this data from this bulk substance, so basically a lot of different types of cells which are taken from alveoles of lungs. They used in the analysis, and then transcriptomics RNA-seq data is performed. And then using this data, the tool is first of all detects actually differentially expressed genes. And then it checks regulatory regions of those genes, which are shown here by gray, gray bulbs around regions which are not actually in the gene, but between the genes in the 5' regions, most of the cases, or in introns of those genes. Those regions of genome are actually regulating expression of those genes. And our tool will take information from that regions, and then using machine learning approach, it will build a combination of transcription factor binding sites in order to understand what combination of factors are regulating those genes. And then on the second step, it will take these transcription factors and will build a so-called network of signal transduction pathways, which are potentially activating those transcription factors. And with that, it will be able to find so-called master regulators in the system. So basically, those few molecules in the system which regulate activity of all these changes of the genes. So we will compare the diseased patients with a normal, non-disease, and then this master regulator will be proposed, which actually control this activity of the gene. And once these master regulators are found, we look in our databases to check whether there are known drugs which can be used to inhibit those master regulators. So that's how algorithm works. And in the core of that algorithm, what we do, we actually try to find transcription factor binding sites in the regulatory regions of genes. So for instance, if there is a transcription factor called AP1, which is responsible very often for activating of genes. So using known positional weight matrix, or so-called a generalized consensus of the binding sites for this factor, which are stored in a database called transfac, we apply these matrices to find new potential binding sites for these factors. And once we found them in a different gene, we can say this, that this transcription factor potentially regulates this activity of this gene. So that's the easy thing. But the more complicated is actually to find which transcription factors work together. And for instance, this is a case of interferon beta gene which is activated by a region in the 5' region of that gene, which is actually target for several transcription factors, which are binding to this region, and they also interact with each other, so-called enhancer-sons. So, and then when there's several transcription factors like RAF and NF-kappa B and this CJUD and CFOS, components of AP1, when they come together, then they regulate activity of that gene. So our goal is to find those places in genome which can be target for multiple transcription factors for such clusters, and which can actually activate the genes, so-called enhancers. Basically enhancers are the regions which may activate the genes through action of multiple transcription factors. And we have inside the tool the machine learning algorithm, genetic algorithm, which finds those enhancers. And so basically doing it for several hours in the computer, try to find the best combination, which is regulating our genes. And at the end, it gives this combination of factors which may regulate activity of our genes. After that, when we find these transcription factors which are regulating activity of our genes, as I said, we start asking question whether there is a master regulator which can activate those transcription factors altogether. And signal transduction network, which is stored in our database called TransPass, helping us actually here to find these master regulators. So we're doing a so-called network analysis. So by finding shortest passes in this network, we're actually getting these master regulators. But actually in the disease, and we will talk about this in the more even in case of COVID, it's a very important issue actually, that in fact there are multiple positive feedback loops which are happening in the cells, which create a so-called new master regulators. So, and the thing is, can you hear me now? Hello? Yeah. So these new master regulators are actually activating activity of these transcription factors. I would say these pathogenic master regulators, right? But the point is that they retrieve the signal from as a feedback loop. So basically they activate activity or expression of genes which express these proteins. So they activate themselves. And while these positive feedback loops are acting in the cell, this pathological actually process is getting under no control. It's getting more and more active. So these kind of feedback loops are really important in, and then we in the algorithm we try to find them and use this as a actually possibility to identify those pathogenic master regulators. And once we find them, then those master regulators which are found by the system can be perfect drug targets, because if we inhibit them, then we can block these feedback loops and then block this pathogenic activity. And the genes themselves, and actually which are encoding these proteins, can be perfect biomarkers, which tells us which kind of pathogenic pathway is which actually switched on. And in case of inflammation, which we actually observe here in a COVID patient in the very late stages, actually is, in fact, is it like a fire. And as you know, fire is itself a feedback loop. And here, these feedback loops are getting very important to catch. And just to introduce a little bit into biology of the process after infection of the COVID. So you know that at the first place after infection, the innate immune response is active, but then later actually the adaptive immune response is getting active. But the problem is when the load of the pathogen is very high, this adaptive immune response can get out of control, and we think it's getting out of control because of those positive feedback loops, which leads actually to a so-called cytokine storm, which in fact then leads to this respiratory distress syndrome, which is often observed in COVID patients. And the major players of these, cytokine storms are the cytokines. And actually there are a number of cytokines which are shown here in this picture, and some of them are pro-inflammatory, some of them are anti-inflammatory. And actually, we have to check in our data which of the cytokines are getting active in this transcription profile. And the system is actually checking it. And depending what kind of role these cytokines are playing, it detects their role as master regulators. Okay, so with that, we can go actually online and show how this all works. So let me this Genome Enhancer tool. So it's an online tool which can be opened just by giving this URL. And then, so I will go widen the screen actually, yeah, so you can see it full screen. Good. So here in this drop file here box, so we are going to put our data, and then this tool will help us to go through several steps by clicking next, and then we'll see how this works. So what kind of data we are going to analyze? Basically, first of all, we got this paper from a Chinese group which published they published microdata of 2 patients, and they store it in their database called Big D, or Big Database. So where they are also transferring this data, I guess, into the major databases of storage of microarray data. But currently, it's still located only in their servers. So they gave access to everybody. Because it's very important to give now free access to all this data. And here, as I said, this BALF, RNA-seq of BALF is done for 2 patients in 2 replicas. So first is for first patient 1 and patient 2. In patient 2, replica 1 and replica 2. So basically we can take this data by clicking here, and taking these FASTQ files by taking this link, copy link address, and then we can go to our tool and actually put this link address here and click upload. So now these FASTQ files are uploading into the system, and of course it takes time, so we'll see how it progresses. But second data is actually were published already in the form of counts. So this patient number 3 actually was published as a count data. So here the Excel table with this data from patient 3. So again, 2 replicas were done, and then there 2 healthy biopsies were done as a controls. And I must say, actually, another difference between these two approaches is that in the first paper, they took actually BALF for analyzing our expression of the data for the 3rd patient. That was a biopsies of the lung. So it's a different sample, but we can analyze it anyway through our system and compare the results. So when we have such data in tabular form, we actually can simply drag and drop it here in our box, and it's actually uploaded, and then we have to import it into the system. It now detects that actually it's a transcriptomics data, and the data is located in the starting from the 2nd row. So it's ensemble IDs in the 1st column. So we are getting doing importing of that data now into the system. So until it's imported, so actually we can go back to the system. So actually the FASTQ files are of course very big. I would say we will stop loading because it will take more than 1 hour. So I have uploaded those data already, so don't be in panic. So everything already in the system, so we can in fact see the data already inside the system. So okay, we go next. And now we have this data loaded. So I loaded this data before. So here, this data for this 3rd patient already loaded into the system. And the FASTQ files, which were from the 1st paper, are loaded in a different folder. So I can show you where they are. So basically, these are this is a data, a lot of files which were generated, but for us, what is important is FASTQ files. And as you remember, there were 2 patients and with 2 replicas. So patient 1 was this one, this 94, and patient 2 starting from 96. So to start the analysis, we have to take this data simply and drag and drop it into this gray box. So we take FASTQ file from first replica, we take another FASTQ file from second replica, and then we take, so that will be the first patient. And the control of that patient was, controlling that experiment was done also, and here are the FASTQ files for the control. So we put actually the data from the control here. So you see, we simply take FASTQ files and drag and drop them here. And also for the, so this will be first patient. Now the second patient will go as well. So basically I will take, where it is, this FASTQ file, we take it here, and this FASTQ file we take here. So this is the second patient. And the third patient is, as you remember, was in the table. So we take the whole table and drag it here. And it shows the columns of that table, giving us information that the counts are in those columns. So now this data are ready to proceed. So we press next. And now we have to create our experiment and control. So let's put the first data here. So for the first patient, we put it here. So, and for the control, so we put the control files. Good, so that's the data ready for the first patient. So then we press next. And the only thing we have to do now is actually select what we compare. We compare the experiment versus control. And now we have to select the diseases we actually want to focus on. And of course, we are focusing now on virus diseases. So first of all, coronavirus infection, and generally RNA virus infection and virus diseases in total. So that covers actually diseases which we are dealing with in terms of virus, but now we also want to look at this data from a point of view of response of the human, of the host. So, and as we know, the response is actually severe acute respiratory syndrome. And also we can put here another information. As you know, a lot of drugs were repurposed from such diseases like another type of infection, for example, from HIV infection. So we will put here this HIV infection in order to understand and in order to find the kind of similarities in terms of drugs with HIV infection. Also, it's known that there is also repurposed of the drugs from hepatitis. So we can add more of those disease terms in order to broaden our scope, how we want to select drugs from that kind of area. And basically that's it. So now we have to simply start the analysis. And the analysis is started and prepared for creation. And then it will take around 4 or 5 hours until all the data will be analyzed, the FASTQ files will be aligned and pre-processed, and then differentially expressed genes will be found, and then all these steps like detecting of master-regulators will be performed, and then detection of drugs will be done. So all this takes time, of course. So we will not wait now for 4 hours. So I will simply go to ready reports which were done with this data. So the reports were done in a different project, this COVID big. And so you can see that there are a number of reports were generated, and then when the process is finished, the system generate two types of reports, so-called brief tale and full story. And full story contains all information about this study. So we will get now to these reports where we analyzed the first patient. So it was actually here. So first and second patient slide. So we will read full story. So the system analyzed all the data, and it suggested these two master regulators as most promising targets. And it also found that these genes, which were found as differentially expressed, are actually controlled. The activity of this gene is controlled. Genes is controlled by these three and these most important transcription factors. And let's go more in details of that analysis. So as you see, first it detects differentially expressed genes. And this log-fold change for those genes are quite high. So really, the number of genes observed very high level of differential expression in this patient data compared to the control. And the statistical significance is very high. So the number of upregulated genes, significant upregulated genes is around 5,000 and downregulated around 7,000. Really a big difference. So then the system actually is doing number of mapping and comparing to gene ontology term, also comparing to classical pathways. So for instance, here it checks whether those upregulated genes are actually changing particular pathways. And as you can see, for instance, pathways related to P53, to apoptosis are related, touched here and activated, and also pathways related to NF-kappa B, so and TNF alpha pathways really activated in this patient. So also in terms of diseases, actually in this patient, a lot of genes which we found upregulated, they known to be biomarker for actually interesting diseases. So diseases related to digestive system, although it's, you know, it's from lung. So a lot of strange genes are activated there. And also a lot of genes which are, you know, which are related to neoplasm and cancer. So actually processes of cell cycle, of proliferation are really get out of control here. The same is done for downregulated genes, so we will not stop here for downregulated, but let's have a look at the actually composite modules. So those clusters of transcription factor binding sites, which were found here as you know, overrepresented in promoters of upregulated genes. As you can see, these clusters contain transcription factors such like FOS, so this is component of AP1, so the transcription factor we were talking about, transcription factor like SMAD, OCT4. So these factors are have something to do with regulation of cell division and proliferation. So we can have a look actually how those binding sites for transcription factors are distributed in the promoters of those genes. So you see, for instance, these are the genes which were analyzed. So we can sort them by the scores. So basically for the number of binding sites which were found in the promoters of those genes. And these binding sites, the clusters of binding sites are shown here in different colors. So you see that they are really found in clusters and they may organize these composite modules. Okay, so then knowing these transcription factors, we're going to the next step and try to identify the master regulators. And this shown here on this network, which is built by the system. So let me zoom in a little bit in this network. So in this network, what we see is at the bottom of the network are those genes which are differentially expressed, which are upregulated in this patient. And as little boxes are those genes. And these genes are controlled by these transcription factors, which we already had the look. So you remember this SMAT, OCT4. So these transcription factors, they are also shown here in blue. So these transcription factors are regulating activity of those genes. So they actually lead to the upregulation of those genes. And upstream, above all those transcription factors, we see this network, signal transduction network, which may regulate activity of these factors. And at the top of this network, you see these pink molecules like PDK1, PDC beta, and this complex of TGF beta, which actually going down. But what's going up is like c-KIT, and those which has this red aura around actually upregulated. For instance, as you can see here, interleukin 8 is upregulated and found here as a master regulator. So the pink ones are master regulators. And among the master regulators is RBB1 actually. It's a epidermal growth factor receptor. So definitely, again, the story is about uncontrolled proliferation of those cells. You see also Aurora A and other important players of the cell cycle here. So from that list of master regulators, then the system gets further, and it takes them and tries to check whether in our database, which is called Human PSD, we have information about drugs which can inhibit those master regulators. And it make a list of these druggable targets, list of targets which can be proposed for this particular patient. And as you can see here, among the important targets, we see here interleukin 1 receptor 1 and some kinases, which definitely play a role here. And next table is actually the final table of the report, which shows potential drugs which can be applied to these targets, to inhibit or activate to work with those targets. And at the top of the list, we see PEG interferon, which is definitely used in early stages of disease, because as we know now, the interferon response is pretty late in the disease, which is different from many other types of viruses. And by giving interferon, it's definitely getting better. So the system found those PEG interferon number 1 because we found those receptors for interferon alpha in the list of our targets, and because actually there's a list of known clinical trials which are going on with this drug for those diseases we selected. So if you remember, we selected the virus, HIV infection, and hepatitis and other virus diseases, and this drug is used against those infections. But then after these interferons, there are other drugs, and to see them, we have to see the full table. And and we have to, it's sorted by this drug score. So, and now we see what other types of drugs are proposed here. And as number 2 after interferon, we see this drug called sunitinib, which is definitely a drug which is used in in cancer therapy, but it's already known, and some clinical trials are done for in the virus, different types of virus diseases, and I will show later that actually there is information going on about use of those drugs, actually anti-cancer drugs, in the case of COVID-19. So as you can see, we can go further with the drug list, but let's have a look at back to the report. And just to let you know that in this report, we also have at the end a list of chemical compounds which are also predicted as potential drugs for inhibiting the drug targets we found. And of course, these compounds are yet they're just for research purpose. But what is done here, we apply a chemoinformatic tool called PAS, which predicts for chemical compounds from a big library of active chemical compounds. It predicts potential drug targets. And it intersects these potential drug targets for these compounds with our target, for the targets for the given patient. And it gives a list of those most promising compounds which can be used here in experiments and further experiments. But let's have a look now to the second, patient. So let's have a look. Where do we have it? So we have here the first patient. We just have a look. And for the second patient, we have the system generated another report, and it actually found a slightly different mechanism. And so I'll jump to the drug list for the second patient. And now you see a different set of drugs, which are proposed here. And at the top, we see here actually antibiotic, monocycline, is proposed as number 1. The second is again sorafenib. And the third is anakinra, and that's a drug which is known for rheumatoid arthritis. And but here it is proposed, and it makes a lot of sense. So I will show later that there is a data now showing that these kind of drugs, which are working against inflammation, has a lot of potential for treating COVID-19 patients. Generally, all these 3 patients give different results of drug targets and respective drugs. And to compare them, we can briefly look at actually comparison results. And for that, we can switch to the more expert view of that system, which is called geneXplain platform. And in that expert view, we actually can do and see more of those data. So here we can go directly to the data projects and actually open the data. Interesting, I'm getting something on the screen. So somebody got access to my screen. You see some hackers are very, very good, I guess, among my people who are listening to me, but I think it will not disturb. Anyway, so you see this data about the FASTQ files, and here we can see also the results of all the resulting tables which we got. So for instance, here is result 3. We got the data about the first patient, and the list of drugs. Here we compare 3 patients together, and we can compare a list of drugs by doing simple Venn diagrams. So here I show this result of such one diagram, and you see there's a list of drugs coming from first patient and the second patient, where the data were taken from both, and the third patient is a biopsy. And you see the number of drugs proposed for the third patients is quite different from those drugs which are proposed for these 2 patients, but there is a number of drugs which are common for all 3 patients. And we can also have a look on those drugs which are common for all of them, so rows present in all 3 tables. And so among them, actually, we can see this combined information for all 3 patients, and we can sort it according to the drug rank for each of the patients and see which position of this drug is in this proposed table for each patient. So so these common drugs are actually proposed for all 3 patients, but the order, the rank of them in our output table is different. So it's actually, we are all different, the patients are different, and these suggestions which the system gives give an idea which drug can be more potential, has more potential for this particular person than the others. So the same as was done for the targets. And so you can see the Venn diagram of the targets. And here is interesting that if you look, for instance, for the targets which are common for the first patient and the second patient, but different from the third patient, as you see that we see a difference in the case of third patient. Maybe it's more later patient in the disease. So what we can open it here, and among the targets, we actually see this interleukin-1 receptor, which I mentioned here in my report as a very important target, which was at the top of the list, is actually also only found for the first and the second patient, but not for the third patient. So it obviously gives an idea that this interleukin, which plays a major role in interleukin, in cytokine storm, is a good target for anakinra, for instance, and it's good for the first and the second patient, but maybe not so good for the third patient. So as you can see with this type of analysis, we can do much more. So we can apply many different methods here to analyze our data, to compare, to intersect tables, to do further studies of the functional analysis. So that's maybe a topic for a separate webinar, how to use this powerful expert system which is behind the Genome Enhancer. But now I will finish with going back to my presentation, and then we have a bit time for questions. So as I promised, basically so concerning those target drugs which are from the field of tumor, of cancer therapy, there's a definite there are recent papers showing that they have a very good potential because actually they potentially inhibit those kinases which takes a role in endocytosis, first endocytosis of the virus in the cell. And by doing that, and in parallel, many of those drugs actually have an inhibitory effect against inflammation. So although they were developed and they are developed as targeted drugs for the cancer, they may be repurposed here for the case of COVID-19. So with that, I would like to finish my presentation. And so here, actually names of our products, Transfag, the database which was used to find the potential binding sites in regulatory regions of genes. Transpass is a database which was used to build these networks and find master regulators. And the Human PSD is a database which was used to find the drugs and clinical trials and to compare and to put this whole information together. So thank you for your attention, and we have now time for questions.
Okay. Now, so first question. Are there statistical information associated with master regulator analysis other than diagram?
So
master regulators, statistics about master regulators besides diagram itself.
Right, so it's a very good question. Thank you very much. So I actually will come back to this picture maybe to give a short answer. Actually, what we are doing here is we are applying the shortest path to find those master regulators. And as you know, this network is huge. It contains thousands of molecules and interactions between the reactions between them. And in that network, there are some hubs. So those molecules which have a lot of connections. And as people search for shortest paths, will always end up with those hubs. In order to add statistical significance, to our search, we actually apply a certain scoring schema. We calculate score for each master regulator, and this score is proportional to the number of transcription factors it can regulate, and it's actually downgraded by a number of other transcription factors which are not involved here, which can be also receive signal from the master regulator. This is score, so which will balance between sensitivity and specificity. In addition, we do actually the bootstrapping of that network and checking if this master regulator can be obtained just by random chance. And we have some p-values and z-scores calculated for each master regulator, and then a certain filtering procedure filters out those which can be found by sheer random chance.
Of course, you can also show this in the table, the report.
Yes.
All the statistical parameters.
Yes,
shown
exactly, there. Exactly. So, so we have here in the report the results of the network analysis. Some of the statistics are shown here. So, for instance, here. So it calculates the scores, which can be, which is, as I said, is a combination of the sensitivity and specificity. Actually, this table doesn't show the p-value, so to get it, you have to apply a special filter. So, but it also can calculate these p-values, and it calculates the p-values and takes into account in filtering.
Okay, the next question is about control. Could you please tell again which samples were taken as controls for 3 patients?
Okay, that's a very good question. I think you had a very good eye watching our presentation. So let me go back to this schema. So in this first paper, which was done by the group from US, there was one patient, and they took actually samples from the lung of that patient. I think it's post-mortem samples. And as a control, they got samples from healthy post-mortem patient. So not healthy in the sense, not infected by COVID-19. So that was, control was also RNA-seq from another patient sample from lung. And in case of this paper from a Chinese group, there were 2 patients, and again, lavage was taken from, infected, but alive, so not post-mortem, alive patient. And the control was lavage was taken from actually another study. It's a study which was done some years before in completely different cohort. Some healthy patients or healthy people were used, lavage from the healthy lungs were taken as a control. So this control were taken actually from another study.
The next question. The main aim of software is finding master regulator and based on that make a decision on which drug is more suitable.
Exactly.
This just yes is the answer. Right?
Yes, exactly. So it's a software again, so which takes expression data. From expression data, it tries to find master regulators, and then from master regulators, predict which drugs are suitable for particular patient.
How are the drugs ranked?
Okay, so the drug ranks, it's a very important question. Let's go into details of that. So ranking of drugs is a not easy task. So let's open this table with the drugs. We have to take into account many parameters. So first of all, so if it is ranked actually by this last column. Let me make it a little smaller so I can see it on your screen. So this last column is a so-called drug rank. So if we sort it by drug rank, so the drug rank should be small. So basically small drug rank. So the drug rank 10. It's a sum of ranks, which are sum of 3 ranks, which are generated by 3 different criteria. So first criteria is how important are the targets which these drugs are using, inhibiting. And the importance of the targets are actually calculated on the basis of another 3 criteria. So importance of the targets is calculated based on the position as a master regulator, the expression of this gene, and actually what else? So whether it was not down regulated, so expression. So then this so-called target activity score is calculated for the drug, which is using this number of targets and the importance, right? And based on this target activity score, we get the first ranking of the drugs, which is called target rank. Then the second rank is done based on disease activity score. And disease activity score is taking into account whether this drug was used for the diseases we selected. So, and remember, we selected as diseases viruses and also SARS, so the severe respiratory acute syndrome. And based on those diseases, it checks whether there are clinical trials which are going on for these diseases, for these drugs. And 25 means that a lot of clinical trials are going on for these diseases, for this given drug. And the higher the score, the better is the rank. And then the final rank is sum of these 2 ranks, plus there is another rank which is called clinical validity, which is a simple thing. It's whether this drug was actually FDA approved, then it's 1. If not yet to any disease, it's 0. Then basically this also taking into account. So in this table, we have not only ones, but we have zeros, and then basically those drugs can be also used if nothing else is found.
Next question. Can we use this tool for another disease?
Yes, of course. And in fact, it was initially created with a challenge of cancer. So if you go now back to the platform, to the Genome Enhancer view, so you see here the demo projects where we applied this tool for different types of cancer, like colorectal cancer, where input was just VCF files, so basically mutations which were obtained in a colorectal cancer samples. Then esophageal squamous cell carcinoma, transcriptomics, FASTQ files, lung cancer, osteosarcoma, ovarian cancer, so different types of cancers. But not only, actually, this tool can be applied to practically, well, to many diseases, not practically all diseases, but to many diseases. For instance, here you have example for diabetes. We have actually cases when we apply this tool for inflammatory bowel disease, to rare diseases, or exactly to diseases, rare disease related to actually problems in the immune system, also to neurological diseases, for instance, Parkinson's diseases, Alzheimer's diseases. So we have a broad spectrum of diseases where this tool can be applied.
Mm-hmm. The next question is actually related one. Maybe you already partially answered to this, but maybe you would like to give more comments. Do the initial format has to be FASTA only?
No. Thank you. And actually, here we have a list of acceptable file format. And the power of the tool is that it's multi-omics. So the example today, which I showed, was just transcriptomics only. But in fact, this tool is able to integrate several types of omics. And you can see here those omics which this tool can work with. namely transcriptomics, epigenomics, ChIP-seq, genomics like VCF I already mentioned, so basically sequence of genome, exome sequencing for instance. Also metabolomics as input can be considered, and proteomics. So, and these are the formats, FASTQ we were talking today, different types of preprocessed FASTQ files like BAM, for ChIP-seq, also BET, so means the position of the peaks. For the transcriptomics, we can work with different types of microarray data like Affymetrix, like Agilent. In that case, you can upload cell files from Affymetrix or raw files from Agilent or Illumina. Mentioned VCF for genomics, or just a list of SNPs, like these RS accession numbers from SNPs can be also input of that algorithm. And the algorithm tries to actually combine all this information from different omics together in one logical scheme so that each data supports each other. So that the answer, so you can apply many different types of formats.
Mm-hmm. The next question. This analysis is similar to other tools like IPA. How is it different from IPA? If it's fine for you, I will start. Alexander, I'll start to make my comments to this question, and then you will help me with additional details, because this is quite can be quite a long story to answer this question. So Genome Enhancer is a unique tool developed by geneXplain company, and we have to mention this. The basic concept behind is concept of upstream analysis, which Alexander presented on his slides. And our concept of upstream analysis includes two steps. First is promoter analysis, and second step is pathway analysis, upstream of identified transcription factors. And I think this approach to upstream analysis is absolutely unique to our approach by geneXplain. Then the first step, this analysis of promoter sequences or gene regulatory sequences for transcription factor binding sites, is done with analysis of DNA sequences, with search for potential binding sites and even combinations of binding sites. And this is very unique, our approach. First, transfac matrices are used here, and this is our unique collection of transfac matrices. And this is done on the basis of analysis of DNA sequence, and other tools are not doing this. Even if they analyze transcription factors, maybe they apply different approaches. Then we are going to networks upstream of transcription factors to find key regulatory molecules. And here, this network behind each and every reaction, maybe you put us this schema, just one schema to screen for everyone to see. Each and every reaction in the schema is taken from transpass database, which is manually curated over many years, starting from bio-based time and then presently at geneXplain. This is transpass database, which contains a big number of reactions and pathways manually curated. This is the basis for our upstream analysis, and therefore this upstream analysis is absolutely unique. You cannot get the same results with any other database which is used behind IPA or any other tool, analysis tool. And second here is, of course, algorithm of network analysis to find such regulators which can, in a coordinated way, regulate the number of transcription factors. This algorithm is developed in our company. It is a unique algorithm which was published already in bioinformatics in 2006. It was slightly modified and optimized afterwards, but basically it is the same algorithm, unique and developed in our company. And the next part here, Alexander mentioned that actually cheminformatics tool PASS prediction of activity spectra for substances is used here for prediction of chemical compounds. And this is very unique feature, and this is definitely is not used in any other tools in this way. Also, we have to mention from the very beginning all this input format which Alexander showed. I do not think that other tools are able and allow to users to, in easy and compatible way, to work with so many different raw formats. Many other tools, they start usually with pre-calculated tables. Of course, people have already their means where they make calculations for differentially expressed genes, but still for many people it is really very comfortable to have everyone together and do all this here within one platform and have actually everything within one platform, and this can be used further on for many additional analysis behind this automatically done part. You can make additional Venn diagrams and compare tables and many other things. So this is roughly about our unique approach and how we are different from other tools. Can we shift to next question now?
Maybe I can just
okay.
I just add a little bit to that. Basically, the major difference is in the title of our tool. It's called Genome Enhancer, and those enhancers is a really unique feature which we are doing. So basically, the general thing is many other tools, including us, in fact, using in the analysis the literature data. So those facts about gene expression regulation, so which are published. When somebody published that this particular transcription factor regulates that gene. But actually, all amount of published information is only a little 1% from what is actually there in genome. So when we limit our predictions just by published information, we actually we are searching under the light. We actually we are searching only those things which are known, and we are losing a lot which is unknown. And using our prediction tools, enhancer prediction tools, we are able to find those things which are not yet published. We find those target genes which are not yet published. So no other tool can do that.
Okay, now we can talk more about this, but let's shift to the next questions. If two people got the same gene expression, does it mean they offered the same drug? If two people got the same gene expression?
Okay, all right. Well, there is never the case, I would say, when exactly the same gene expression is observed. Even the same person, if you take a second sample, it will be a certain variability of expression due to technical noise, due to noise, well, kind of noise in the system, because also different cell types are expressing in different ways in different time points. So you will never find exactly the same profile. But when two profiles are quite similar to each other, then the tool will predict similar drugs. So it's a kind of deterministic in a sense, right?
Mm-hmm. The next question. Target activity score is based on what?
Target activity score, okay. So target activity score is based on targets. And let me go back to the targets. Let me check. Okay, so if you go to the table of the targets, somewhere here. So transcription factors, targets. So as you remember, first step is finding the master regulators. But then we consider not all master regulators, but only those ones which can be considered as targets. So basically, when there are already known drugs for these targets. And then what is done, these all targets are sorted by the total rank. And as I mentioned, this total rank takes into account several things. It takes first into account whether this gene encoding this target is highly upregulated. The second thing, whether this gene is actually at the top of the pyramid, if it's capable of regulating transcription factors. So by combining these two types of ranking, we calculate this total rank. So each target has its total rank. Now the drugs using these targets. So basically, when we have consider a particular drug, we check which targets this drug has. Then we go to this table of targets and calculate some kind of target activity score. So basically, it is you can read this formula in our report. It's summarizing with a particular weighting coefficients of those target ranks. At the end, the higher the target activity score, the higher, bigger numbers, so we can actually sort by this target activity score. You see this bigger activity score is assigned to those targets which have a biggest importance in this network. But that's not the only thing we have to consider. Although these targets are so important, but there are no drugs available in the market for those targets. That's why at the final drug ranking, we include also a number of clinical trials for these drugs. So we try to really compensate or balance knowledge about the targets and knowledge about the drugs involvement in this disease.
Next question. The blood plasma proteome can be integrated into the pipeline?
Yes, very good question. In fact, in this paper, which we're analyzing data from, they did not only lavage, but they also did RNA-seq, not from plasma, but from PBMCs. So actually from cells, macrophages, which were filtered out from the blood. So, and we also analyzed this data, so this may be another topic to discuss. And in the PBMCs, in other types of cells, you see different picture. And in fact, the whole picture of response of immune response has to be considered from a point of view of many cells which are involved here. And in that sense, lavage is in fact a somewhat good example because it's already a mixture of many types of cells. So in one sense, it's a bad example because it's given mixture signal, but in a sense, it gives the actually integrated response, and it leads us to maybe a good predictions at the end without decomposing all this into several types of cells. So the answer is yes. So we can try to incorporate different types of cells or different types of liquids, where we can take this information from, like from plasma, from whole blood. But then the system becomes much more complicated. So at the moment, we prefer actually to work with integrated system. But more and more data are coming now from a single cell transcriptomics and also on COVID-19. Our goal is we starting analyzing also single cell data. And maybe in some next webinars, we can present data from single cell analysis when the sample is disintegrated in different subtypes of cells, and each subtype can be analyzed through this pipeline, and then at the end, results of different analysis is integrated in one response.
The next question. Is the data set searched against human and virus proteins?
No, not yet. Not in this system. So Genome Enhancer is working with human only, and we are not considering the virus proteins. Actually, the only possibility to consider that is to take into account data on protein interactions. And there is a data now, there is one paper recently published where people did actually proteomics, protein-protein interaction between proteins of the virus versus proteins in the host. So they listed the host proteins which are found to be interacting with the virus proteins. So this type of information can be taken into account in our analysis, but not the proteins of the virus itself.
Mm-hmm. The next question, is there any approach to validate the accuracy of drugs that takes higher ranks by this software?
Yes, sure. The only and the most important validation would be experimental validation. Mm-hmm. And basically, of course, it's the clinical trials on people would be difficult to establish with such pure, computational predictions. But still, there are, since now is a big need for such information. We know that some clinical trials, for instance, I know the information from Israel, where they starting now clinical trials based just on computational predictions of drugs, because they need such information, and that's not something impossible.
Maybe we can add here that actually system suggests also drugs that are already approved by FDA.
Yeah, each individual drug is approved by FDA for a different disease. So of course it's not known whether it will be working for this particular disease, and especially in such complicated situation like modulation of immune system. Mm-hmm. So, but so the final validation is, of course, on clinical trials.
The next question. We have data on 3 conditions, and we want to find genes which are upregulated in condition 1, comparing to both condition 2 and 3. Mm-hmm. Next, we want to find master-regulators for these genes. Can we perform such analysis with Genome Enhancer?
Yes, let me briefly show to you. So at the final step, when basically we have to, let me go to that. So when you initiate the analysis, then you can here put several conditions. So what I showed to you is like patient versus control. But you can add more conditions. So, and you can say if I have patient 1, patient 2, I have control, and then I have to say, okay, this condition is actually my baseline. So this control, and then the algorithm, what it will do, it will compare routinely. It will take first condition, compare to baseline, make a report, takes another condition, compare it to baseline, make another report. At the end, you will have all these comparisons in one report. Yes, mm-hmm. You can do that.
Okay. The next question. From which database do you find transcription factors for expressed genes?
From TransFac. Well, actually, not, again, so TransFac is used to find potential binding sites for transcription factors in the target genes. So we use positional weight matrices. So as I showed in my presentation, so-called, more extended variant of consensus of these binding sites for transcription factors to search in potential regulator regions of those genes, and then even to search for clusters of those binding sites. And then through that, we predicted which transcription factor regulate which genes. So we are not taking it from any literature information, we're predicting it.
Thank you, Alexander. I think that these are all questions for today, and we already have taken more than 1 hour. We had so many very interesting questions. Thank you all for your time and for so active participation.
Yes, right. Thank you very much. Yes.
With this, we would like to finalize for today, and please, you are welcome to send us further questions by email.